Good morning, students. In today's lecture, I will be discussing about the phylogenetic relationship between different groups. While we are considering the concept of homeobox, as I have already discussed in previous lectures, that there are specific sequences in a DNA. These sequences code for certain proteins. These sequences are called the homeotic genes. The subset of homeotic gene is called the homeobox sequence gene, and within this homeobox sequence gene there is a very conserved sequence of DNA, and this conserved sequence of DNA, which in case of Hox gene is 180 base pairs. This subset of homeotic gene is called the homeobox. And as already discussed, during the evolution, the organisms, be it simpler form of living thing or more complex, all of them have the same conserved sequences in their genes. And this phenomena where we see the similarity between various organisms, simple organism to complex organism, this similarity in terms of gene is called the unity of type. One view suggested by Etienne Geoffrey Saint Hilaire and Richard Owen was of unity of type, which suggests that all the organisms have some common ancestor from which various organisms, complex organisms, have originated. And they called this common ancestor between the invertebrates and vertebrates as a bilaterian ancestor. But Charles Darwin, along with other thinkers of that time like George Cuvier, Charles Bell, etc., they focused on the differences among species that allowed each species to adapt to its environment. For example, the hands of human, the flippers of the seal, wings of the birds and bats were such examples which showed that the animals have adapted to their condition of existence. Thus, the two views regarding the evolution and the development of organisms called, one view is called the unity of type, and the second view is about the condition of existence. The difference among species that allowed each species to adapt to its environment is called the condition of existence. Adaptations were secondary, and that the unity of type was critical. By unity of type, we mean that the organisms have similar structural organization. They have some ancient, some common ancestral gene. For example, the various homeotic genes like the Parkes gene, the Tin Man gene, and the tailless gene, as well as the Hox gene. All these genes, they have a homeobox, which suggests that all these genes have some conserved sequences within them and which is present in simpler animal as well as in higher animal. So this is known as the unity of type, where we find the homologies in different structures. As mentioned, hands in human, flippers in seal, wings in birds. So all these exhibit the homologies. Similarly, the evolution consists of modifying the embryonic organisms and not the adult one. This was suggested by Metchnikoff in 1891. The unity of type or descent from common ancestors, and for that, as I have mentioned, a common ancestor was designated to be as the Bilaterian ancestor, which was said to be a common ancestor between the protostomes and the deuterostomes. The condition of existence is equal to the natural selection. When we say that condition of existence is there, it means that the different organisms belonging to different phyla, they have adapted themselves according to the environment in which they are inhabiting. So there is a common descent which shows the embryonic homologies. When we examine the embryos of human or various animals, we see and we know a very important theory that ontogeny repeats phylogeny. That means the development of an individual, when it is occurring, we see that during the development of individual embryo, it repeats its phylogenetic or previous ancestral characters, like the presence of gill cleft, like the presence of tail or the remnant of tail in form of tailbone. So when an individual, for instance a human individual or human embryo is developing, ontogeny means the development of an individual. So during its developmental journey, it repeats the phylogenetic or its ancestral characters. The modification shows how the development was altered in order to produce the structures that enabled the animals to adopt to a particular condition. When we say modification, we mean those changes which an individual, which an organism has undergone in order to adapt itself to a particular situation. To find out the underlying unities that link different groups of animals, we have observed this while we studied the homeotic genes. Several genes, they show unity, the underlying unity between various groups of animals, and to detect those differences in development and that enable the species to adapt to particular environment. Now when we say that there exists a unity of type, all the organisms, they have similar plan for structural organization, for the development of eye or for the development of heart. But when we say this, we also know that in spite of having some unity, a wide variation is there in organism. We know a fly is different from a frog, and frog is different from a human, a man. In spite of the fact that there exists a unity in form of the homeotic genes from the ancestral common ancestor, but still there is a differences, and the aim of the topic is to detect those differences in the development, what has occurred, that the body structural plan even in arthropods are so different. When we talk about phylum Arthropoda, we know there are distinct classes like class Insecta, class Crustacea, class Chilopoda, class Myriapoda, class Arachnida. So all these varied classes are there. In insects, we have different type of insects like the uh, ant, we have the honeybee, we have the mosquitoes, we have the housefly. All these insects are different. Then again, the second class, crustacea, we have varied type of crustacean animals like the prawns, the crabs, the lobsters. Even within the same class, there is difference. Within the phylum, there is difference. Why this difference is there? This difference is there because the organisms, they have adapted to their environment. We can say that they have descended with some modification, or we can say that they have conditioned themselves to exist, condition for existence. Then if I talk about the chelopods, we have the centipedes having so many legs. Insects have only 3 pair of legs. uh, Crustaceans, they have many legs, walking legs are there. Then swimmerets, swimming, uropods and pleopods are there for swimming. These are the swimming appendages in crustaceans. In centipede, that is chelopod, so many walking legs are there. In diplopoda, example millipede, number of legs are there. In arachnids like spiders, 4 pair of legs are there. So even within a same phylum, so many differences exist, and this is known as the condition to existence. And the aim of this chapter is to detect those differences in development that enable the species to adapt to their particular environment. Now this is so the view unity of type proposed by Etienne Jeffrey, E T I E N N E, Geoffroy, Geoffroy, then Saint Hilaire, Sinthilaire, and Richard Owens, Richardowen. These researchers or thinker proposed the origin of organism or evolution by proposing the unity of life, which I have already explained. Then the other thinkers, like George Cuvier, Georgecuvier, Charles Bell, Charles Bell, and several others, they have proposed the condition of existence or condition to existence. Darwin acknowledged these two theories, unity of type and condition to existence, in 1859, and he wrote that the two great laws, unity of type and condition of existence, are both necessary to explain the evolution. He went on to explain that his theory, origin of species, would explain unity of type by descent from a common ancestor. Darwin noted that the homologies between the embryonic and larval structures of different phyla provided excellent evidence for descent with modification. He also suggested that adaptations that depart or go away from the type and allow an organism to survive in its particular environment develop late in the embryo. He recognized two ways of looking at descent, DESCENT, descent with modification. One could emphasize the presence of common ancestor or common descent by pointing out homology in embryos. So this is a phylogenetic tree that explains various types of animals. It shows the similarity, or it shows the presence of a common ancestor designated as ancestral protist, which gave rise to porifera and then to other types of varied animals like coelentrates, platyhelminths, mollusks, etc. So there is one ancestral protist designated by yellow circle, which is a common ancestor for all these animals from porifera going up to vertebrates. All these animals have a common ancestral protist. And then again, there is another yellow circle which bifurcates into two, protostomia and deuterostomia. Protostomia having phylums like Arthropoda, Nematoda, Annelida, Mollusca, helminths, and deuterostome having all chordates, hemichordates, echinodermata, urochordate, cephalochordate, and vertebrates. So these are all the deuterostomes. So both deuterostomes and protostomes, they have arisen from a common ancestor known as the bilaterian ancestor, and it is because of imagining or considering that there is a common ancestor, we see that there are certain genes which are present in simpler animals also, as well as in higher animals also. That is, the homeotic genes like the Hox gene, Parkes gene, Tin Man gene, tailless gene, etc. All these genes are the homeotic genes having conserved DNA sequences which are passed on from one animal type to another. If such a similarity or a hypothetical ancestor is there and such a similarity exists, then these are the genes that are present: the Parkes gene, the Tin Man gene, the tailless, all these genes, orthodentical, empty spiracles, all these genes encode for the formation of head. Hox gene, it is present in all the organisms. It specifies the anterior posterior axis specification. So the question is, if the underlying Hox gene expression is uniform, then how did these differences emerged? What might have been the mechanism that vast variation in the anterior posterior appendage appearance, presence of maxilla or absence of maxilla, presence of walking legs or absence of walking legs, presence of walking legs in larval forms like in caterpillar, absence of legs in larval form, example maggots. So all these differences, why it has been there, if a common homology is followed, if a common gene pattern is there, then what is the mechanism behind the differences? We are going to understand that because of the presence of some proteins, the other genes may be switched on or they may be switched off. If because of some protein the other genes are switched on, certain characters appear. If because of the presence of certain proteins some genes are switched off, then those characters do not appear. For example, if because of the presence of Hox protein some genes are switched on, then maybe the wings may appear, and if because of the presence of Hox protein certain genes are switched off, maybe the wings may not appear. So in this lecture we will be understanding the mechanism why among the same phyla and even among the same class, why these differences originate. So we can say that if the underlying Hox gene expression is uniform, how did the differences among the phyla emerged? How the Hox genes are regulated, and what target genes the Hox encoded protein regulates. There